So yeah, thanks again for the invite. My name is Matthew Holt, and I'm going to be talking to you guys today about our work in muscle invasive bladder cancer. So I'm part of the Precision Proteome Initiative here at Baylor College of Medicine, where in the Texas Medical Center, we have a lot of different clinicians with many different cancer cohorts. And our goal is to take these sometimes undercharacterized cancers, do discovery proteomics combined with data analysis, and finally generate potentially clinical assays with mass spectrometry to actually guide therapeutic decisions. So today I'm going to talk about one of our projects, which is in muscle invasive bladder cancer. And what is muscle invasive bladder cancer? Basically, bladder cancer is commonly divided into non-muscle invasive and muscle invasive. And typically it's thought to start at the lumen, so this is the inside of the bladder, and then it gradually grows and becomes more invasive into the muscle, and that's the stage in which is called muscle invasive bladder cancer. About 25% of the new cases of bladder cancer are considered muscle invasive and has an overall fairly poor 5-year prognosis. This isn't the worst cancer, but it's still not great. And the issue really here is that the standard of care is quite medieval, right? It's still basic, just a very simple chemotherapy followed by a radical cystectomy. So this is a complete removal of the bladder. And I guess if you didn't already know, smoking is bad for you, so you shouldn't smoke, but it is also one of the key risk factors for bladder cancer. And bladder cancer is the fourth most common cancer in white men. So of course, there's been plenty of research towards bladder cancer already, but most of it has been in the RNA field. This is sort of like a hallmark study generating RNA-based subtypes for over 400 cases of muscle invasive bladder cancer specifically. They did do protein here, but this is an RPPA array of only 200 different proteins, so it's not very much coverage or depth. But what's important is that they found different submolecular subtypes that are associated with risk and response. So what we designed is a proteogenomic, similar to like what SEPepQuant does, approach, starting off with 143 patients. And as you might envision, muscle invasive bladder cancer has a challenge in that there's a lot of invasion of the muscle, so there's a lot of muscle cells and muscle proteins. So there's obviously two ways of dealing with that. You could take Roman's fantastic approach that he just presented using microdissections, or you could take what we did and simply filter out for the samples that you have, these biopsies that only have very high tumor content. So the caveat for that is that from our initial 143 cases, we ended up with only 46 pre-treatment cases and 14 matched post-treatment cases. And with these, we went ahead and we've done whole exome sequencing, RNAseq, TMT profiling, TMT phosphoproteomics, and CHARACTERIZATION. CHARACTERIZATION is Thermo's term for standard heavy label isotope triggered PRMs. Really quickly, I want to discuss what we call resistant and what we call sensitive. So anything that decreased from a high stage muscle invasive bladder cancer, that's defined as like the T2 plus, to a lower stage that's not muscle invasive bladder cancer, so that's T1, T0, we call this sensitive. And anything that didn't respond to the chemotherapy at all, or increased, or responded slightly, but is still muscle invasive, we call resistant. With this definition of sensitive and resistant, we have a very significant stratification in overall survival. We also see a stratification between sex, so with women having a significantly worse overall survival than men. I'm going to breeze through the genomics real quick. Essentially, with a 10th of the cohort that previous studies had from the TCGA, we didn't identify anything new genomically. However, the previously identified top mutations, we basically recapitulated all of them in this graph. And the tumor mutational burden of bladder cancer is extremely high, and this is sort of like our cohort versus the previous cohort of bladder cancer at a very similar tumor mutational burden. So despite having a small cohort, we're still fairly confident that we can identify the key features that were previously seen. For this, since we're kind of like mimicking SEPepQuant in this approach, and we're also part of SEPepQuant, we're using the Martins et al. TMT protocol here. And this is sort of a fantastic protocol in nature methods and nature protocols. And there is one key issue, I would say, with this protocol, which is in this step, where you have to do a QC to check to see if your TMT labels, if they've finished labeling, so the labeling percentage. And this is very time consuming with our previous setup, where we take 2 micrograms from each channel, you speed vacuum, you desalt, you speed vac, and then you run these on the MS. And this is combining all of these. What we found is that in combining all of these, you honestly can't really tell if there are medium problems with any single sample. And to run each one individually on the mass spec, we were looking at 22 hours of that work. So I'm gonna talk a little bit about using the Evosep to do TMT here. So first off, it's obvious that the Evosep is not optimized for doing TMT. And in this XIC here, in blue, we have one of our standard DIA runs. So this is label free on the Evosep 44 minutes. And then in red, we have a TMT run where you can clearly see that there's been a significant gradient shift. But despite this gradient shift, you still can identify about 70%, at the very least 70% of these peptides, which we think is enough to actually quantify the QC step. And so we took the sample with TMT labeling, and the thing is TMT is extremely reactive. So in only 5 minutes, even if you're using a lower ratio of TMT Pro, you still can see the vast majority of your peptides already being labeled. And so this is in a 44-minute method, we already see about 7% just unlabeled peptides. And so we took this sample and we ran it in multiple different speeds. And what we observed is that we can actually pretty much quantify how much mislabeling we have at very fast gradients, such like the 5.6-minute gradient. I will give the caveat that you do need to optimize your method if you're using an Orbitrap for this, because if you're using a very time-expensive MS1 and MS2, so high resolution, high injection time, you will have very few data points in a 5.6-minute gradient. But if you're using only a single TMT tag per run, you don't actually need the high resolution MS2s, right? So that is a solution if you're using an 11-minute or a 5.6-minute gradient. And we've changed our protocol to incorporate TMT QC using the Evosep. So in this case, instead of taking the samples, drying them, cleaning them up, drying them again, then running them on easy NLC 1200 or whatever, we take the samples and we throw them directly into the Evotips, and then we run them straight into a 5.6-minute run. And this took what used to take a day and now takes basically a couple hours. So this is significantly expedited, sort of like our TMT QC process. Now, going back to the results, comparing the RNAseq data that we got from this cohort and the TMT data, pretty comparable to previous CP-TAC studies. We have a median correlation of about 0.5. This is kind of expected. RNA and protein don't correlate that well. And certain subtypes, so certain classes of proteins, have even worse than the median correlation. And this is commonly reported to be things like, OXPHOS and ribosomal and other protein and translational machinery. And then on the right here, we have a comparison on how well each omic can predict the creation of complexes. So all these different complexes have known stoichiometry, and essentially we expect the detection of the members of these complexes to have similar stoichiometry as predicted. And in conclusion, using the proteomics data, which is here basically in the y-axis, it's better predictive of complex stoichiometry than RNA-seq data. So essentially, proteins better predict proteins, shocker. Now, because subtypes are so important in muscle-invasive bladder cancer, we decided to create our own multi-omic NMF subtype. And this was done primarily with the proteomic and phosphoproteomic data. And that's what you see as these top NMF subtypes here, 1 through 4. Now, these are significantly associated with sensitivity. So for instance, NMF subtype 3 is enriched for resistant samples, while 3 and 4 are also enriched for female samples. However, the previous subtyping, the TCGA subtyping over here, is largely incapacitated by the new NMF subtyping. And this is kind of shocking if you think that the overall correlation is less than 50% between these two omics, yet you still generate the same subtypes. With one major caveat that we are obviously separating this more sensitive TCGA subtype into 2 different subtypes. And this is primarily driven by these metabolic pathways that we identify in this cohort. which really you don't get as good quantification with the RNA-seq. Moving on. So the reason why we still get good correlation between these different subtypes is because generally when you're doing subtyping, you are looking at the most varied genes, the genes with the highest variance, right? And as it turns out, the genes with the highest variance tend to have higher correlation between RNA and protein than just any random gene, which makes perfect sense. So here at the top, we have the overall correlation between RNA and protein. And over here, we have 480 genes that are used in the standard bladder cancer classifiers. And this is a very significant difference. It's actually the most significant difference that we observe between these. So in essence, the current proteins that are typically measured by RNA-seq have very high correlation between each other. And these include very therapeutically relevant ones, such as EGFR, BRAF, GATA3, etc. Now, this isn't to say that there is no new information to be gained from proteomics. In fact, quite the opposite. When we compare the results of what we see by GSEA between the RNA-seq data and the protein data, we see some very interesting differences. So in this plot, on the y-axis, you have the RNA data with anything going up here is associated with sensitive, anything going down is associated with resistant. And on the x-axis, you have the same thing, but for protein. So anything going to the left is resistant, anything going to the right is sensitive. A lot of the previously described mechanisms of platinum resistance are observed here, things such as EMT and myogenesis. And interestingly though, when you start looking at the differences between what the proteins identify and what the RNA-seq identifies, you get things like Wnt signaling, which is not significant at all in the RNA level, but is quite significant at the protein level. And interestingly, KRAS signaling is considered down by the proteomics, but up by the RNA-seq. So you might even get some differences in conclusions as how these cells are potentially becoming resistant or are already resistant to the chemotherapy. The other thing we have is phosphoproteomics data. And there's many different ways of looking at the phosphoproteomics data and normalizing it. In this case, what we wanted to see is that can we detect a phosphorylation change that is happening with no change at all at the protein level. And that's what we see here in GSK3b, where the protein level is not significant changing at all, and the phosphorylation is, however. This GSK3b phosphorylation is important in signaling and is generally considered to be deactivating of GSK3b. And the downstream signal seems to be also elevated in Wnt pathways associated again with resistance. One quick thing I want to discuss with you guys, this is a different way in which we look at proteomics data. This is a structurally equivalent peptide group, SEPEPs. So this is work done by Young Chow. As you guys know, sometimes a peptide can map to a single gene that only in a single protein. Some peptides will map to multiple genes and multiple proteins, while others might map to only one gene, but multiple isoforms within that gene and don't discriminate between any of these isoforms, while others are partial discriminators, and some are fully discriminative and only quantify one gene, one protein. All right, so you can take this and you can make this bipartite graph over here. And when we say structurally equivalent, we mean structurally equivalent within the graph, right? So peptide 3 and peptide 4 are basically giving you the same information. Peptide 5 and peptide 6 are giving you the same information. So we consider these individual SEPEPs. So essentially what we have is a different way of quantifying peptide groups at sort of like the isoform level. So we applied this logic to this cohort as well. So here on the y-axis, we have the gene level. So this is as if you're just doing regular proteomics quantification between resistant and sensitive, where up top is resistant and down low is sensitive. And on the x-axis, we have the SEPEPs. So this is basically the isoform level information, with on the right side being resistant, on the left side being sensitive. What's really interesting here is that we see in these highlighted boxes areas where the isoforms are significant while the protein in general, the gene level, is not significant. And I want to highlight here ATAD1, right, in this SEPEP. So ATAD1 is an interesting protein because its primary role is to remove membrane-bound proteins. And it does this primarily in the mitochondria. However, this CPEP is missing the mitochondrial localization signal and also the transmembrane domain. And so we have two hypotheses here. One, this protein is no longer functioning as it should. And one way in which cancers can become resistant to chemotherapy is by literally pumping out the chemotherapy out of the cell, right? So this protein can remove those pumps. And if it's no longer functional, it's no longer removing those pumps. The other way it could be functioning is that it's no longer functioning in the mitochondria and it's having effects elsewhere in the cell. This is particularly important in the context of ADCs. So what is an ADC? So an ADC is an antibody drug conjugate. And simply put, this is a missile that people have created where you have an antibody that's targeted towards a specific antigen or gene on a protein, and attached to it, you essentially put a payload. So this is often just a chemotherapy. And this allows you to have a much more focused and much more targeted approach of delivering chemotherapy to a cancer cell and killing both it and neighboring cells. So ADCs are extremely hot in bladder cancer right now. And here are all the ADCs that we detect, the targets of ADCs that we detect, which are also FDA approved. And in particular, Nectin-4 and TROP2 have shown a lot of promise in clinical trials, as well as PD-L1. What's interesting is that Nectin-4 and TROP2 have a very high correlation between the two of them, right? So if you just look at their distribution here. However, PD-L1 has a very poor or negative correlation with Nectin-4 and TROP2. So our prediction here is that the combination of PD-L1 inhibitors with a Nectin-4 and a TROP2 targeted therapy is going to be better than the monotherapies by themselves. And interestingly enough, there's actually an ongoing clinical trial that's actually testing this. So it's only going to finish in 2026, but I'm excited to see if this hypothesis pans out to be true. Going back now to the isoform level quantification, I want to briefly mention looking at families of proteins rather than an individual protein, and how sometimes looking at a protein family can have better statistical power than looking just at each individual member. So in this case, I'm highlighting these RAF family members, so ARAF, BRAF, and RAF1. When you look at them individually, they don't have particularly strong p-values. They're not the biggest changes. Some of them are not significant at all. However, this particular CPEP is taking a region, specifically the kinase domain, out of all of these genes, and looking, if you just look at these peptides, which are shared between them, how does that look? And in essence, that's the strongest significance between resistant and sensitive that we see. So this is essentially an enrichment of a domain rather than an enrichment for a specific gene that we observe as being relevant. And this makes a lot of sense because they have highly analogous functions. So perhaps a tumor wants to have multiple of these functioning at the same time, or is just choosing one or another, right? So it's just a different way in which looking at isoform level information can benefit. I want to switch real quickly to talking about targeted proteomics here. So in this case, it's internal standard PRMs that are sort of like the end goal of what we want to do with this. We recently just published this paper looking at kinases, where we, very much like Kuster, have a series of inhibitors that we've conjugated to beads, and then we use this bead cocktail along with lysates to enrich for kinases, and then we run those on the mass spec, right? And kinases are really cool to look at because they're druggable, and a lot of them have either FDA-approved drugs, just not indicated for this particular cancer, or they're currently under development for new FDA drugs. And in this assay, our final output was 106 heavy peptides targeting 80 kinases. And we showed that in these patient-derived xenografts, 16 of them, when we do this kinase pull down, we can be highly quantitative in subtyping and identifying which kinases are elevated in which cancers. So we apply this methodology. We're going to apply this methodology to our muscle-invasive bladder cancer cohort. But there's one thing I wanted to highlight real quick about using the Evosep for these type of analyses. So in our hands, when we load 1 microgram, that's measured by nanodrop, This is essentially saturating the evotips. And we really find that the linear range of the evotips are between 100 to 800 nanograms. And this is actually a problem because if you overload your heavy peptide standards, you will see a decreased signal in your heavy peptide standards, which will significantly influence your conclusion on the data. And so what we have done to essentially solve this is just an extra step in loading. So first off, we just process your evotips as you normally would. And then we load just the stable isotope labeled peptides and then run those through. And we don't usually add a wash step here because the peptide solution is already very clean. And then after that, we load our endogenous peptides. We aim for 500 nanograms, but in our tests, actually, you can largely overload the tips at this stage because you already have a fixed set amount of your peptide controls on the evotip itself. But 500 nanograms is our SOP, so we generally stick to that. And so what we ended up doing is the CHERACTERIZATION, which for you guys who are not familiar with CHERACTERIZATION, this is a process by which you have a watch mode. So you have a targeted set list of your heavy peptides that you just added, and you add them in such a high amounts that you can easily detect them. So you can have very cheap MS2 scans for them. So this is like 5 milliseconds per scan to do an MS2. And then you can do a pseudo spectral match real time in the instrument when you detect one of these heavy peptides. And then that triggers the acquisition of the endogenous light peptide in a much more expensive MS2 that often takes, a tenth of a second. This lets you have very high accuracy, precision, and robust quantification of your endogenous peptides. So we took our kinase panel that we had and we applied that to the muscle invasive bladder cancer cohort, specifically because we had both BRAF and RAF1 as peptide targets. And what we see is that we can reproduce the previous quantification that we saw at the SEPep level. We can also see that when we do our heavy peptide controlled targeted proteomics, where these genes are highly associated with sensitivity. So kind of trying to piece a lot of this together, we see BRAF and RAF1 as significant in sensitive cancers. So this little graph over here, anything up is associated with significance, anything with sensitivity, anything down is associated with resistance. So we have copy number, RNA, protein, and then the phosphorylations. So for instance, for RAF1, we can see that it is significantly associated with sensitivity for the RNA data, the protein data, and this one phosphorylation. Meanwhile, BRAF by itself is not associated with anything significantly, except for this one phosphorylation, which is up. Conversely, these guys can phosphorylate MAP2K2, and it's interesting because some of the phosphorylations of MAP2K2 are associated with sensitivity, while another one is associated with resistance. And MAP2K2 can then phosphorylate STAT3. There's also evidence that RAF1 can phosphorylate STAT3 directly, as well as IL-10 receptor, which is the IL-6 STAT3 pathway, is a canonical pathway. We see this associated with resistance, and we see overall STAT3 seems to be a nexus for resistance, where it is significantly down by RNA, protein, and deactivating phosphorylation of STAT3 is also associated with resistance. Subsequently, we also see the downstream targets of STAT3, which include cell cycle progression and growth, are also elevated at the protein level. So this is just a different way of trying to piece out at what stage are we seeing these different genes be modified and modulated in bladder cancer. I also want to talk about how we try to validate some of our results, and also in this case, how we try to come up with new therapeutic hypotheses. So because the standard of care is quite simplistic for muscle invasive bladder cancer, there's a desperate drive to try and find anything new that could help these patients. So one way we have of prioritizing the genes and the drugs that we might want to give to these patients in a clinical trial would be using DETMAP. So this is a CRISPR knockout database developed by the Broad. And what we have here on the y-axis is the FDR of the CRISPR. So essentially, if this gene was knocked out, and this is specific for bladder cancer cells, in the bladder cancer cell, how much did that affect its growth? So if it affected the growth, it's going to get a good p-value. It's going to get a low p-value. And on the x-axis, we have the protein levels significance between resistant and sensitive samples. So everything we have highlighted in this box here was both significantly elevated in the resistant samples, but also we have cell line evidence that these matter when they're knocked down. So these are dependencies for cancer growth in the cell lines. And the color labels here are based on if these genes have a known inhibitor and at what stage of the FDA process these inhibitors are in, right? So you might have a very good gene like LRP1, which seems to be associated with resistance and has a significant effect in the cell lines. However, it doesn't have an FDA-approved inhibitor for even cancer. So this would be sort of like a target of repurposing. And so just for my conclusions here, I think getting this level of RNA and protein at the same time, I think can be very informative as to how these cancers are actually working towards developing resistance or have an innate resistance to chemotherapy. And in that matter, we found that Wnt signaling and STAT3, as well as GSK3B, are particularly important in this. So we're following through by doing a PDX trial where we have a series of patient samples that we've engrafted into mice, and we're testing to see if drugging these genes that we've predicted are going to be effective in treating those cancers. And lastly, I want to thank our funding agencies, BCAN and CPRIT, as well as everyone who's worked on this project, with a specific highlight on Seth, who is the bladder cancer urologist who has largely led this whole endeavor. And with that, I'll take any questions.